Welcome to today's lesson. We'll look at Mendel's experiments more closely, and we'll look at Punnett squares. If you want to learn about genetics or how a trait is inherited, it can be very helpful to take two individuals, have them reproduce with each other, cross them with each other, and see how the offspring turn out. And this is where a test cross comes in. A test cross is a breeding experiment used to determine the genotype of an individual or to determine the inheritance pattern of a gene. A monohybrid cross is going to involve individuals that are identically heterozygous for one gene, not two genes, and breed them together or self-fertilize them. So this is a monohybrid cross because we're looking at two individuals. Up here is the father, and this is the mother on the left-hand side. And they are both heterozygous. They both have a capital B and a lowercase b. Capital B to represent the dominant, the purple color, and the lowercase b to represent the recessive, which in this case is white color. And same thing with the mom. She is also heterozygous. A Punnett square is this diagram, and it helps you predict the outcome of a cross. Even before you cross the two individuals, you can predict the chance of producing a particular type of offspring. For example, I might ask, what is the probability that these parents, given their genotypes, will produce a white-colored flower? And I can predict using this table because I have a 1 in 1, 2, 3, 4 chance of producing a white flower. So I have a 25% chance of producing offspring with a white flower. So Punnett squares can be really useful. They can help predict the outcome of a cross even before that cross happens. So let's do a sample problem. What is the probability that a mother who is heterozygous for brown eyes and a male who has blue eyes will have a baby with blue eyes? So we're going to look for the question and find clues. So we know that the mother is heterozygous, and the heterozygous genotype is a capital B and a lowercase b because we have one that's dominant and one that's recessive because heterozygous means two different alleles. The father, I don't have that information, but I know that he is a male who has blue eyes. So I know that the father has blue eyes. And the only way to get an individual with blue eyes, because blue eyes are recessive, is to have two recessive alleles. So the father's genotype is going to be little b, because if I had a big B in there, then the father would have brown eyes. And since he doesn't, we know that his genotype is little b. Now we can draw our Punnett square. On the Punnett square, we're going to write the genotypes of our parents. So on the left-hand side, I'm going to have big B, little b to represent the mother. And on the right hand, or actually on the top, I'm going to have the little b to represent the father. And this is basically what could be in each egg cell, right, if this was a female, and what would be in each sperm if this was a male, right? So each sperm for the male would have a little b in it, and for the mother, she could have eggs with a big B or eggs with a little b after meiosis. So when we write this, we set up our Punnett square, all we have to do is basically fill in the little squares. So in the first square, I'm going to have a big B, and I'm going to match it up with a little b. In the second one, I'm going to have a big B and a little b. In this third box down here, I match up little b with little b, and in the fourth square, I'm going to have a little b and a little b. So I write in my letters, my genotypes, and then I can determine the phenotype, what the individual looks like. So big B, little b, the big B is dominant, and it's going to win, so we'll have a baby with brown eyes. Same thing in this box. And then down here, I have this individual with two little b's, and this individual will have blue eyes, and so will this one. So my chance in this cross of producing a child with blue eyes is 50%. So this couple has a 50% chance of having a baby with blue eyes. This is how Punnett squares work. Now, Gregor Mendel did a lot of this kind of stuff. He did a lot of monohybrid crosses, and he drew Punnett squares and calculated probability and all of that stuff. And just a quick refresher of Mendel's experiment, particularly with the flower colors. He took purple flowers and self-pollinated them to create true breeding purple flowers. He did the same thing with the white flowers, so we have true breeding white flowers. And then he crossed a purple and a white flower with each other and produced his F1 generation. And this F1 generation is all heterozygous. It's a mix of these two parents. So it's going to be big P, little p if we're using the letter P. By the way, it doesn't actually matter what letter you choose to use. We're going to use P for this experiment. And then he took these purple flowers, these hybrid flowers, and self-fertilized them, producing the F2 generation. And in the F2 generation, this white flower showed up again. So let me show you this on another picture. Here are our true breeding purple flowers, our true breeding white flowers. They crossed them with each other to create the hybrid, the big P, little p. And he took two of these heterozygous individuals and crossed them with each other. And we have them right here in the Punnett square. So we have big P, little p, big P, little p. And when we cross them with each other, we end up with the first box having big P, resulting in a purple flower. The second box having big P, little p, resulting in a purple flower. The third box having big P, little p. Right, big P, little p, resulting in a purple flower. And then that last box is going to have little p, and that would be our white colored. So again, 25% chance of producing an offspring that is white in color. And this ratio kept showing up, this 3-to-1 ratio. Remember, Mendel studied a lot of different traits. He was looking at seed shape, seed color, pod color, and this 3-to-1 ratio kept showing up over and over for Gregor Mendel. Now, to go back to this experiment, the flowers, the F1 plants were all purple, but the F2 offspring, he actually counted 705 had a purple color and 224 had a white color. And if you reduce that, that is that 3-to-1 ratio. And for each of the 7 characteristics that Mendel studied, he found that similar 3-to-1 ratio in the F2 generation. This led Mendel to really think critically and come up with an important law known as the law of segregation. And the law of segregation states this: when an organism produces gametes, each pair of alleles is separated, and each gamete has an equal chance of receiving either one of the alleles. So here's a parent, and this is homologous chromosomes, and homologous chromosomes have sometimes they have the same version of the gene like they do here, so 2 capital Y's, but they could have different versions. One could have a capital Y, and the other could have a lowercase y. And the law of segregation says that each pair of alleles is separated. So you could end up with a big Y here and a little y here, and there's an equal chance that each gamete will have one or the other of the alleles. So that means in these homologous pair, the alleles can get separated from each other. And that led to a lot that we know and use for our Punnett squares. Now, Mendel wasn't finished. He tried to do a second round of experiments where he decided, you know what, why don't I not look at one trait? What if I look at 2 traits at the same time? This is a dihybrid cross. It involves a cross between 2 individuals who are identically heterozygous for not 1, but 2 genes. So let me show you what I mean. So let's say we look at 2 things. We look at the outside of the pea plant and we look at the inside at the same time. So the outside of the plant is called the husk, and it could be green or yellow. And the inside of the plant is the seed, and it could be, again, green or yellow. For the husk, the outside green color is dominant over yellow, and for the inside, yellow seeds are dominant over the green seeds. So this is what Mendel did. He took a true breeding, a homozygous dominant plant, right? Which is big G, because we're going to look at 2 traits now. So that's why there's 4 letters here. So we have big G to indicate the green outside. And then let's say these seeds are yellow. I know that I tried to make them look yellow, but they are. And the yellow is dominant on the inside, so we're using capital Y, capital Y. So that's this plant, true breeding to be green on the outside, yellow on the inside. And he looked at a homozygous recessive plant, which is going to be the recessive color on the outside, little g to represent that yellow. And the recessive color on the inside is green, so we're using little y to represent that. So this is the genotype for this individual. He then decided to cross these 2 plants, the homozygous dominant and the homozygous recessive, with each other. And if we're looking at the allele combinations for the first parent, so what would end up in each, let's say, egg of this parent, each egg is going to have a big G and a big Y, right? We're going to cut this info in half, and each egg is going to have a big G and a big Y. That's the only combination. And for the homozygous recessive plant, let's say this is the father, all of them are going to have little g, little y. So when we cross them together to create the hybrids, we're going to end up having an individual that has a big G a big Y, and a little y. So this is what Mendel did. Took his pure green on the outside, yellow on the inside, and pure yellow on the outside, green on the inside, crossed them together to create this hybrid. And the hybrid, of course, looks exactly like the dominant parent because that dominant allele is masking the recessive one. But these offspring are hybrids. They have a secret lowercase g representing the yellow pod color and a little lowercase y representing the green color. So then Mendel took these plants, these hybrids, and crossed them with each other. So he decided to cross the two hybrids, the big G, big Y, little y, and cross it with another with the same genotype. Now the allele combinations for the first plant are these. You could have a big G, big Y. You could have a big G, little y. That's right here. You could have a little g, big Y, and you can have a little g, little y. So these are the combinations, the versions that you could have for your egg. Right? Your egg could have this in it, the egg could have this in it, the egg could have this or this. And the allele combinations for the second plant are the same. So when you do your Punnett square, you're going to put each of these combinations into your Punnett square up on the top and here on the left. And then you just cross them in. You literally just write the letters. You do G's first and then the Y's. So I have big G, big Y, big Y, and then you can determine the phenotype. So big G means it's going to be dominant color on the outside. Dominant color on the outside is green, and the dominant color on the inside is yellow, so it's going to have a yellow P. And then what Mendel noticed when he did this Punnett square, when he did this cross, actually he didn't do the Punnett square, he did the cross itself. He crossed the P's together and then spent all this time counting, is that 9 of them were green on the outside, yellow on the inside, which is the double dominant. So 1, 2, 3, 4, 5, 6, 7, 8, 9 that were that color. There were 3 that were green on the outside, green on the inside. There were 3 of those. 3 yellow on the outside, yellow on the inside. And then there was 1 that was double recessive. So recessive for the outside, recessive for the inside. And that's this one, yellow pod color, which is recessive, and green on the inside. And this 9-3-3-1 ratio kept showing up. Just like the 3-to-1 ratio showed up for the monohybrid cross, for the dihybrid cross, it was 9-3-3-1 that kept showing up for Mendel, no matter what he decided to do. If he was looking at purple and yellow, purple and white flowers, or tall and short plants, he kept seeing 9-3-3-1s. Okay, this led Gregor Mendel to come up with another law, the law of independent assortment. So in all his experiments, Mendel noticed the 9-3-3-1 ratio. And this law, independent assortment, states that when gametes are formed, the alleles of each gamete segregate independently. So basically, when they line up in metaphase, the alleles can go different ways. They segregate independently from each other. So different alleles and different gene types can go separately from each other. So basically, in the example that we did, the seed color and the pod color, the color of the husk, segregate independently each other. It's not like green pods always have green seeds. Green pods can also have yellow seeds because the 2 different genes segregate independently from each other, resulting in this variety of offspring, or this variety of the daughter cells, the gametes rather, the eggs and the sperm. So linkage groups. Genes that follow Mendel's law of independent assortment are either on different chromosomes or they are far apart on the same chromosomes. And all the ones that Mendel looked like were just like this. They were either on different chromosomes or they were really far from each other and they segregated independently. If genes are far apart, crossing over occurs so frequently that they can assort into gametes independently. Linked genes, which we'll look at closer later, are genes that are located close together on the same chromosomes and that can't separate from each other, so they're likely to be inherited together. They are called linked genes. So I hope this was a helpful introduction to Punnett squares, monohybrid, and dihybrid test crosses. Thank you so much for listening.